The overall goal of this approach, which combines an impedance-based real-time cell analyzer and a high-content screening-based platform, is to provide information on effective doses, toxicity, and biological effects to obtain the tox profile of the tested compounds.
This approach can help answer key questions in the field of toxicology, addressing, for example, cytotoxicity, genotoxicity, and cellular stress responses. Moreover, not only provide information about multiple biochemical and morphological parameters simultaneously, it can also be applied to a different range of cell types and cell culture models. Due to video restriction and limitation, the entire approach will be performed while assessing only a short list of compounds. For the same reason, only one protocol will be shown for demonstrating the high-content screening assessment. Demonstrating the procedure will be Stefano Acali and Alexandra Laurent, technician from my laboratory.
To seed the normal human bronchial epithelial cells, or NHBE cells, program the instrument to define the number and duration of impedance measurements. In this study, data is recorded every 15 minutes for 48 hours. Measure the plate background by pipetting 50 microliters of pre-warmed medium into each well of a 96-well RTCA plate. This step represents a technical requirement for the instrument to calculate the medium electrical resistance to use as a baseline reference for cell-based calculations. Next, add 50 microliters of cell suspension at a concentration of 144,000 cells per milliliter to the medium already in each well of the RTCA plate.
Once the cells are seeded, it's very important to leave the plate at room temperature for 40 minutes.